Dear students, very warm welcome to this first class of the basic genetics course. So, this course will be taught by three of us, me, myself, Richa Arya from Department of Zoology, Banaras Hindu University, along with me, Professor Madhuji Tapadia and Professor J.K. Roy will also teach you various aspects of genetics during this course. So, let's first start with this class where I'm going to introduce you with some basic fundamental concepts of genetics, and I'm sure that you are already familiar with most of them because of your 11th and 12th education, and you must not be unfamiliar with several of the terms. In this class, I will help you go through with these old concepts that you already have, and then I also add on some concepts to get you to a place where you can understand the advanced concepts of genetics. So let's move on. So this is our week 1 and unit 1. In this unit, we are going to mainly focus on elements of heredity and variation, right? So heredity is something where we are talking about passing on different characters from one generation to another, from parents to their offspring. We will talk about it in more detail. And then what are the elements of variation? What does that mean? Means how different type of variability arises. What is the basis of that variability, right? Like you will see when we will discuss Mendel's experiment where he talks about tall plant and dwarf plant, or yellow seed or green seed, and so on. So those are the variations. So what is the source of variation? How those variations arise? So before going into the detail, it is important always to understand the historical aspects. Like if we start discussing about Mendel's work in this unit, we should also know what people know before Mendel started his experience, that help us give a clear perspective, like what are the questions that were unknown at that time and that people were trying to understand. Why I'm saying that this kind of thing is more important now when we have advanced so much in genetics and genomics actually, that some of the things they sounds very obvious to us. But then when Mendel was working, or when these people were working, probably more than hundreds of years ago, what exactly their system, what were known at that time. So it is important to get the correct perspective. So in this few slides, we will be talking about the foundations of genetics. So let's talk first about what is genetics. I briefly already introduced you that genetics is a branch of biology where what we study inheritance, we study interplay of genes, like how genes interact with each other. We also study how genes actually are being influenced by their environment. So genes are being expressed. We all know that they make proteins and they are various important components that are required for the functionality of an organism. But that function of a gene is also modified by environment. So there are regulatory elements, there are expression, there are several other things that are regulated by environment. So all together, all these things, like how traits are passing from one generation to another, how the genes are interacting with each other, and how the environment influence these gene expression, it comes under the umbrella of genetics. So this is just a cartoon to show you that it's not something that Mendel did not know, or maybe people before them did not know, that traits, rather I should say characters, pass from one generation to another. We get several of our features from our parents, and people used to recognize them, and we are being recognized based on those features, that we are daughter of so and so, or son of so and so. So this cartoon is just to show these two traits where there's the father is having like the long nose, and mother is having curled hair, and the daughter is actually getting a long nose as well as curled hair. So traits are, what I'm trying to say with the help of this cartoon, is the traits, they pass on from one generation to another. That was known from ages, right? People also knew that they can hybridize, to closely related species, or organisms, closely related ones or closely related breeds, and then they can get better breeds, right? So this phenomena was also known. People use it for crop improvement and animal improvement. But what is missing then? The missing aspect is how exactly this kind of variations are being transferred from one generation to another. Where are from these variations coming? Where exactly they are located? What is the source of variation, right? So these are the things that people did not know. So I would say, in a more specific word, that systematic approach for quantitative analysis is something that was missing. So the phenomena is something which is already known, but how it is happening is not very much clear. So before Mendel, you see, Mendel, we will discuss about Mendel's work 1865, when he publishes some like sort of 1800. But before 1800, there was this theory which is the preformation theory. Preformation theory, what this theory says? This theory says that probably there's a whole organism present within the sperm or a egg, right, as this is written here. So what they were imagining, that how the trait are transferred from parents to their offspring, because in the sperm or the egg every small little detail is already present, and then those details are being transferred to the kid. But this theory later on was disregarded, because when the microscopy was invented, I'll discuss a bit about it, and then embryology was discovered, then this theory did not stand correct. So they dismissed the theory. And so what I'm trying to say is that before Mendel was this kind of theories that we already know that transmit from one generation to another. People were trying to propose different theories to explain what could be the possible reason. And Mendel's work and its rediscovery, why I have included both briefly here, because Mendel has given postulates, we'll come to it, and then later three different scientists understood, they were working on their own independently, and they also learned that Mendel already have done some part of it, and they understand from Mendel's postulate, they bring out the laws, right? So the Mendel's discovery actually highlights two major things. And what are those things? That the characters, they transfer from one generation to another, and they are discrete. They don't mass up with their identity. We'll talk about it. And then two independent characters do not influence each other. So Mendel's finding largely can be classified in two major laws. Law of segregation. So in law of segregation, Mendel used one character at a time. We'll discuss it in a minute. And under the law of independent assortment, Mendel used multiple characters to show that multiple characters can also pass on from one generation to another, and they do not influence each other. So again, it is also important to know what sort of advanced knowledge at the time of Mendel was available to Mendel. So you can see these slides actually highlight the important discoveries that happened in 1665. You all are familiar with Robert Hooke, who coined the term cell when he looked at the cork sections and he found cell-like structures there, repetitive units like there, and then he called those repetitive units as cell. And 1833, we learned about the nucleus. So Robert Brown found that every cell is having this nucleus, right? So nucleus was discovered. And 1858, like just before Mendel, Rudolf Virchow also said that cells are important for life. So one cell is formed by the previously existing cell. So continuity of cell theory, that was also being proposed by Rudolf Virchow. So that was the information already available to Mendel, right? And around the Mendel's time, it's not that when Mendel was working on understanding of heredity, he was the only person, as I just said, that it was a question so many people were interested to know about it, and many of them were very critically pursuing it. And so no other than a known person like Darwin. He was also working on the understanding of heredity. So you remember that Darwin has a book on origin of a species a few, like about I think 10 years or so back, before he proposed this pangenesis theory. So in origin of a species, Darwin, with his all experiments and voyage, discovered how the evolution play an important role in origin of a species, right? So that is the important concept, and he was trying to understand how exactly that is happening. So he proposed a theory which he named as pangenesis theory, and what he considered that there are tiny particles that are known as gamules. They are shed by every cell of the body, and these particles actually mix during fertilization, and that is how they passes from one generation to another, right? And the caveat of this theory is he was thinking that even acquired characters can also be, he was hypothesizing, and he was thinking that even acquired characters can also be inherited in the same way. So the theory was eventually later rejected, but it is important for you to know that even the timing of Charles Darwin and Mendel is overlapping, right? And then other person who is Francis Galton. So Galton was the cousin of Darwin. So he was very close cousin of Darwin, and he was working around the same time when Mendel was working. Although Mendel dies very early, but the duration of their work is very similar, although they were not really aware of Mendel's work. So that was something which is very odd. Probably now we are living in an era where things are electronically communicated and communicated very well, and so we know about any discovery very quick, but before probably it was not that easy. So working in the same time, people were having different approaches to address same question. So Francis Galton was also trying to understand heredity, and Galton and of course Darwin knew each other, and they were discussing, and they were in the same time frame. But Francis Galton was trying to understand heredity by using human traits, right? So he was considering the intelligence and the fingerprints to understand how these traits are transferred from one generation to another, right? So we'll talk about it, but his focus was the continuous trait, right? Eventually we'll discuss what was the strength of Mendel when he chosen his organism to study and the traits to study, why he come to the conclusion where other people could not draw those kind of conclusion at his time. So selection of model organisms, percival, how you are designing your experiment, is also very important criteria to get the correct answer. So in coming back to the Galton's work, he was looking at continuous traits, and then he was also following rigorous statistics to understand his data, but he found that these things blend very smoothly. Whereas in case of Mendel, we know that he was using discrete traits, and he clearly saw the traits do not get mixed up with each other. So, Galton actually discussed more about nature versus nature, and then he said that heredity is more important over the environment, and he proposed the concept of eugenics, which is also very controversial, but we are here to appreciate the idea of scientific pursuit, how people are pursuing same question from different angles, and then we want to understand up to what extent they were really able to solve a particular problem. So now coming back to the main focus, right, we all want to know more about the work of Mendel, what exactly he did, how he concluded his data, and what we learned from him about the heredity. So just a very brief outline of Mendel, that we know that he is known as father of genetics. We will see why he is considered as father of genetics. One thing that I would like to say, that sometimes some discoveries are way ahead of their time. So Mendel's discovery is amongst one of those. When it was discovered, it was really difficult to possibly understand the depth of that discovery, and which actually eventually, with more data, more understanding, repeated thinking, helped us understand what exactly Mendel has already told and showed with the help of experiment. So Mendel was born in 1882. As you can see, I have listed few important facts on the slide. He was born in 1882 in Austria, and he was always interested in nature and science, right? And he came from a very poor family. His father was a farmer. He wanted him to be a farmer, but Mendel wanted to pursue his own passion. So he joined the monastery in Brno, and in this monastery actually he went to study physics and mathematics. So you would be amazed to see that Mendel was, while he was working in the monastery, he sent out for his studies, and whom he was working with during his studying period was Christian Doppler, the Doppler who was known for the Doppler effect in the case of sound waves, right? So he was working under him, he was working under Franz Unger, and Franz was somebody who was using microscope to study the organisms, right? So that is what his background. It's not that he was just a monk and started working on pea plant and got some postulates, but he was having a very good background and a lot of interest to pursue science with passion. So then after his studies, he came back to the monastery, and then he started working there. He was giving the teaching position, and he was teaching to the secondary school students, and along the side he was working on the scientific questions that that he wanted to address. So he published his first paper. It's important thing for you to know that he published his whole work that he did mostly in about 8 years on P in 1865, and actually unfortunately he died very early at the age of 61. I personally feel that if he would have lived longer, probably nobody would have need to discover him. He would have been known because he might have been presented his work multiple places, and people would know about his work probably a bit earlier than he was discovered later in 1900. So let's come to what exactly Mendel's experimental setup was, right? So as I said that the two things that he discovered, major things that he discovered, how he came up to that idea. So he was working, I'm sure that you all know that he was working on pea plant, the Pisum sativum, and this plant is good because it has several traits which helped Mendel to see and analyze his data clearly. So what I have listed actually here, that these traits are easily observable. You can see here. These flowers are bisexual. So pay attention to this clause that I have written here. There are several things that make pea plant a very good plant to study the kind of things that Mendel wanted to study. So these plants are bisexual, which means that they have male and female part in one flower itself, right? And the structure of plant is such that the leaves, petals actually closes in such a way that it won't allow the pollen to come and go so easily. So automatically the plant prefers self-fertilization, right? And then if you artificially cut the stamens, you can actually allow the cross-pollination. So you try to understand that the system is flexible. If you need pure things like self-crossing, as we come and discuss in a minute, then this automatically the plant structure, flower structure allow you to do so. And if you want to do the cross-pollination, you can also do it by manually interfering with the pollination process. So Mendel selected 7 traits. We will talk about what traits he has selected, and he worked for almost 8 years, and he did several experiments. He meticulously recorded the data and analyzed it. So I would say that again, one thing that doing experiments is not only important. The important is also how we look at data, how we analyze it, how rigorous we are in our data keeping and data analysis. Then only we are able to come to some conclusions, right? And so he also applied statistics to understand his data better. And we'll talk about it slowly in our coming slides. And as I mentioned before, all the work that he has done on the pea, he published this work in 1865. So these are Mendel's character and traits. So see, one thing that I want to mention, there are several words that Mendel used differently than what we know today, right? Because now we have better understanding, we have better way to unambiguously use terms without any confusion. But for all of us, it is important to know what terms Mendel was using. So what I have done is I will keep on highlighting those terms while I teach you, but I have also made the reflection of those terms towards the end so that you can revise it if it is needed at any time. So you can see Mendel used the word character, you can see here, and he has used the word trait, right? So what are the characters? So characters are like flower color, flower position, pod shape, plant height, pod color, seed shape, 7 characters, right? So these are different characters that Mendel took. Now, whenever we are talking about a character, there could be many variations in that character. There could be one character at, that is it. But the character could have more variations, 2 variations, maybe 3 or even more. So here for the simplicity, we are talking about 2 contrasting variations within this character. So you see the flower color, let's take the first example. Flower color, what he is talking about, purple and white. So a flower can have different colors, and so what word he used? So he said flower color is a character, and in that character there are 2 traits. The flower is of purple color, one trait, and flower is of white color, another trait, right? So just follow this. I'm sure that you must be aware of the concept of gene and allele. So when he is talking about traits, he is talking about alleles, right? So different form of a gene is allele, and these alleles, because of some changes, they may also have some observable changes like the flower color. So the character, eventually you would see he has used the word factor. So he has used the word factors because factors are being transmitted from one generation to another, you will see later. So factors are actually genes and traits are actually alleles, right? So you can see that there are many different traits in a character category that he used to study. So remember one thing, we'll talk again, I will repeat it in my class, but again one of the strength of Mendel's work is that he used the contrasting traits. Within a character, he used contrasting traits like purple flower and white flower. He used tall plant and short plant. He used round seed and wrinkled seed, something which can be very clearly, cleanly seen and observed and recorded. That was again a strength of Mendel's work. So Mendel's methodology, again there are a few more terms, a true breeding plant. So what he was doing, he was constantly using true breeding plants and hybridize them. We'll talk about it. And what he was doing, I already said that he was very meticulously recording and quantifying his data, and whatever data he was having, he was also statistically analyzing that. So with that, I talked to you about some strength of Mendel's work, what was his sort of research planning, and so I would end my class here. So with that, I already talked to you about some aspects of Mendel's work, that what kind of plant he has chosen and what are the different traits and characters that he has used to study the heredity. So with that, I stop here. In my next class, I will talk in more detail about Mendel's methodology and his discoveries, different experiments that he has performed, and then what sort of conclusion he has taken out from that. So with that, I want you to thank all of you for your attention and for your time. I hope that this class would be helpful for you to at least understand what was the scenario and what was the kind of research happening during the Mendel's time and what was Mendel's approach to approach the questions of heredity. Thank you.